Welcome into the Inside the Ice House podcast, our second annual AI Biotech series. Today's guest is Jason Kelly. He is the CEO of Ginkgo Bioworks. Jason, thanks so much for joining us inside the Ice House. Happy to have you here.
Yeah, happy to be here, Lance.
So at its core, when you and your co-founders decided to start Ginkgo Bioworks, and I should mention we're obviously talking here on the day of the AI Bio Summit at the NYSE, when you were thinking of starting Ginkgo Bioworks, what problems were you all trying to solve, and what is the ultimate vision that you're still working towards this day?
Yeah, so I started Ginkgo out of MIT. So I did my PhD in bioengineering at MIT, and my co-founders did too. We met there. There's 5 of us. I don't know if you have had a chance to work in a lab at all in your life, Lance?
Oh, I have not. I was a journalism guy.
Okay, well, school. You're lucky. Okay. So it turns out you want a PhD at MIT in bioengineering. It is 5 years of standing at a lab bench, very carefully moving clear liquids around, trying desperately not to make a mistake, setting up your experiments.
Well, your precision must be just like phenomenal.
No, it's horrible. In fact,
you must have very steady hands.
Yeah. Yeah, they'd say that though. Yeah, actually, some people in the lab are known to have, quote, good hands. Okay. All right. That's like a thing.
Were you one of those people?
Absolutely not.
No. Okay.
Yeah. And so that little bit leads to why we started Ginkgo, right? So our view was, if you look at bioengineering, and I can talk a little bit about even what that means, but come back to it. Okay. The limiting reagent into it, like what slows it down, is the lab work.
Got it.
All right. And we believe that you can take a lot of that work and automate it so that scientists can spend their time thinking and just order what experiments they want from the lab bench. Yeah. All right. And so we could either then commercialize that by selling you the robots, or we could be like an AWS running it for you in the cloud. But the idea is you should yourself stop doing that manual work and free yourself of the bench.
And so you bring up this engineered biology.
Yeah.
Where do you think, well, you can first get into it, what is engineered biology? And then once you do, where do you think sort of engineered biology has the greatest opportunity to create real world impact?
Yeah. So, all right. I'm a product of Jurassic Park.
Okay.
So that book came out when I was 13 years old. So I'm sort of the second generation of biotech people, right? Like Michael Crichton writes that because Genentech is out and these first generation biotech companies making these drugs. And he sort of jumps forward and says, well, what if we could really just design DNA?
Yeah.
And I think the thing, like, to help put a point on it, if you think about the technologies we have in the world that run on code, there is exactly 2.
Okay.
Computers and cells. All right. In computers, it's zeros and ones. In cells, it's A, T, C's, and G's. Yep. But everything else, like if you look around this room, this table, the chair, none of this other stuff is running on code. Yeah. Okay. It's just like the phone in your pocket and you. Got it. All right. And so what's amazing about cells compared to computers, and by the way, like running on code is very powerful. So if you download a new app on your phone, new code goes in, and suddenly magically, like, you order a car, or you can talk to it, and it can make the same thing do lots of different stuff. That's the power of like a code-driven product. The limitation of computers is they only move information around. So if you look at what they've disrupted, right? Media, finance, telecom, things like that. What have they not disrupted? I don't know, like hamburgers and building materials and fuels and, all the atoms-based things. Well, a cell is programmable like a computer, but it doesn't move information around. Biology moves atoms around. So the reason I get excited about bioengineering in the long run is it's sort of our atom compiler in the way the computer is our information compiler. And we're in the extreme infancy of using biotech. And it's so early that we basically equate it today to developing drugs, because that's the first app that's come out on top of, in my opinion, like the matter compiler computer of a cell. Well, the only thing we've really been able to make money doing with it yet so far is disease treatment. But if you think of it in the long run, it should be touching every physical goods industry if we actually got good at programming these things.
So you say we're in its infancy.
Total infancy.
What does the process look like toward getting out of the infancy into, if you equate it to baseball, middle innings?
Yeah, sure.
End of game, 8th, 9th, maybe even extras.
Yeah, let's get to the 3rd inning. Okay, so this is a great question. So let's say you want to program a cell.
Sure. All
right. So example would be like a yeast cell like you would use to make beer. This is like a very easy thing to do. Sure. You learn to do this as an undergrad in school.
Super easy.
Super simple. Just pick up a pipette and get to work. So the way it works is the cell has a genome.
Okay.
All right. So like a bacterial cell, for example, has like 1 million to 3 million letter genome. You have like a 4 billion, 3 billion, whatever letter genome. All right. And so you identify what you want to add to that cell. All right. So let's say it's like a brewer's yeast. Sure. Let's say you want to add a gene. The very first drug product was human insulin. Okay. So you could look up the gene for human insulin, go on your computer, type the ATC GGGG or whatever of the gene, hit print. The DNA gets made. So we can chemically make any DNA sequence you want now. It's called DNA synthesis is the technology. It gets made. And then have you heard of like CRISPR?
Yes.
Okay. So what CRISPR is, it's actually pretty simple. It's basically find location in genome, cut, open. All right. So now you have gone to a place in a genome, and there's other tools that do this too, whatever. CRISPR is only used for certain things, but you open the genome and then you stick in your human insulin gene. Plus you've added a little thing on the front called a promoter that turns it on. And then you close it all back up and put the cell in a tank in South San Francisco in 1978. You grow it up and you start making human insulin for the first time, where diabetics used to get it from pigs. And so that's like the simplest thing. I've just taken a cell and I've made one little change, make this protein for me. Does that make sense?
It does.
All right. Here's the problem. Why are we in the first inning and not the third? Humans did not design cells. All right. This is the key distinction between computers and basically everything else, cars, planes, the whole shebang, and bioengineering. It is the one discipline of engineering where we are working with basically alien technology.
Got it.
Okay. Right. Like not invented by humans. Does that make sense?
Yes.
And so it can run our code. We can put code in, but you don't know what half the parts do. It's like you had this computer landed from Mars or something on a meteor, and you could put a disk in it, but you had no understanding of the operating system or anything. You couldn't read the code. There's a bunch of zeros and ones. It's like, would you like to write some zeros and ones and put them in? And so we've been essentially trying to figure out how cells work for the last, 100 years. And then since, the late 70s, early 80s, we've been able to insert our own code and be like, how about this? How about this? Yeah. But most of the time it doesn't work because we didn't design them. Does that make sense?
So it's that lack of knowledge, essentially?
Well, now let's talk about, so that is part of it.
Okay.
And that's half of it. Okay. So there's basically two ways we get to the third inning. One is we greatly improve our knowledge of the cell.
Okay.
And a lot of what you're hearing at the conference we're doing today at New York Stock Exchange is, could I have an AI model that understands a cell?
Sure.
Right? Like, could I put the whole thing inside a model and then now I could simulate it? Like I can simulate an airplane or like I could simulate a semiconductor chip. I think that's great. I think people should try. That's like one approach. That's what you just suggested. Improve our knowledge.
Sure.
The other one is, we can try today. Like we work in the lab and we stick some DNA in and we see what happens. It's trial and error.
Yeah.
By the way, this is kind of how we write software, right? We don't simulate Microsoft Windows or something. We let a bunch of people get in there and go at it, right? It's like it's high throughput trial and error. That's how we write software. Maybe it's like that. That's how our other coded thing works. So could we reduce the cost? Because now this is why we're not in the third inning. I just said, hey, make that DNA. We pay by the letter. So imagine if you were writing a new piece of software and you paid for every 0 and 1.
And it didn't work. You just wasted all that money.
Yeah, it's expensive. And so what we are doing, like our bet in this whole thing, is if you reduce the cost to compile and debug, essentially to run through cycles in the lab, we can also get to stuff that works through like more rapid trial and error. We also want to be smart about it, but let's just be honest. Software benefited from the fact that you could have like monkeys at keyboards just banging away and it would go quick, right? And you could just try. And if it didn't work, it didn't work. And you added debuggers, you had all this stuff. Like we got to get there with bio, but that's the Ginkgo bet is we can solve it on the lab side. A lot of other people are also trying to solve it on the knowledge side.
And so where does in all that autonomous labs fit in? Because I know that's a focus of Ginkgo.
100%.
Where do those fit in the greater journey of getting to that, third inning that we were talking about?
Yeah. So then if you now start to say like, well, why is it so expensive to compile and debug genetic code? Is it that the chemicals are expensive? Like the A's and T's and C's and G's, are they expensive? Actually, no. We get them from like In fact, we get them like salmon sperm or something insane. Like you go collect this stuff and there's not a DNA in the environment. And right. So you can get these chemicals pretty cheaply. The reagents at low volumes for everything you need to do aren't that expensive. The equipment's expensive, but actually you can do a lot of throughput on a piece of equipment. It turns out when you really dig in, the expensive part of the compiling and debugging are two things. One, the technicians, like the people doing the work in the lab, and two, the labs themselves. Like you have regulated space, right? Like you have a BSL-2 laboratory, people are wearing lab coats, you need air changes, like it's got to be safe for the humans in the room. And so those are your two big costs, it turns out, which is crazy. But when you really get down and you look at it like an engineer, that's the dominant cost. And so we believe with an autonomous lab, and the idea behind autonomous labs like an autonomous car. All right. You sit in the back of that Waymo, you're like, hey, Waymo.
And you let it go.
Take it to my house. And it does it with no human driving. Same idea here. You could sit outside my lab in Boston, go on, in this case, like go into Claude Code or Codex, say, hey, I want to run this experiment. We have like a harness, like a plug-in that goes into that. It translates your request into code on my robots, and it just runs without a person in there.
Sure.
Okay. That's an autonomous lab. And that starts to get you on the path to making this stuff way cheaper. And once we do that, then there's a million and one things you can do. You can miniaturize it, 50 other things. But like we're still in the era where we're still like using, if you want to use the computation analogy, like slide rules. All right. Like we're manually doing our compile debug.
So you compared it to Waymo, right? Yeah. And I think with autonomous vehicles, or you compared it to autonomous vehicles, you mentioned Waymo. So I think with autonomous vehicles, there is still hesitancy behind them.
Sure.
Right? People trust them more, I think, than they did, say, 5 years ago, or 3 years ago, or even a year ago.
Yeah.
But like if I got in a autonomous vehicle, I'd be a little scared.
Have you been in one?
I've never been in one.
Oh my gosh.
I've never been in one.
Try one, you will not be scared after.
So okay, so that's fine. So you say that. You say that.
Yeah.
Is there still that feeling at times with autonomous labs?
Oh yeah. We're early on in the adoption of this. And the real question is less like it's the big challenge in our space, not to nerd out too much, is like with a Waymo, it's how do you handle the variability of the environment?
Sure. Right?
What if it rains? What if you go on a dirt road?
What if there's a detour?
A guy rides in front of you on a bike? It was all these questions of like, can it handle the million edge cases as you move through physical space? Okay, that's actually not our problem with autonomous labs. I can totally control the environment. I'm like, nothing, no one's allowed in, nothing changes. Like I have control over the space. What I don't have control over is what experiment you asked for. So it's really the variability in the requests that you make that I have to handle. And so the scientists are all curious, can you do my experiment? Like that's what they want to see. Like, oh, well, I have super weird stuff, Jason. Like, could that run on your robots? And so it's just a process of like letting them try it. We offer it as a cloud, so you can go to cloud.ginkgo.bio and order an experiment. We can talk about this in a minute too, because I think you could eventually open this up more broadly. But today our customers in pharma or in academic science or in ag biotech could order from us, try their favorite experiment, and if they like it, maybe then they build their own lab, or maybe they then order a lot more from my cloud.
What is, or if autonomous labs become more common, what does that mean for scientists moving forward? Like the human manpower of it all?
Yeah, this is super important too. So it's actually good timing. So the director, Michael Kratsios, who heads up Office of Science and Technology Policy for the White House, he's a cabinet member, just put out a report yesterday about the U.S. in a golden age of science. Like it was basically the science plan from the White House.
Sure.
And it is worth a read if you care about this stuff at all, because what they're suggesting is we need to change how we do science in the United States in a substantial way. Sure. And we've been kind of doing it the same way since the 1940s, roughly. Like people at benches, the NSF funds you, professors, the whole shebang. And the argument in that paper is roughly like, that's not competitive with China anymore. So if you look, there's great scientists in China where it's, this is no longer China 25 years ago where they didn't have like talented scientists trained in their own academic institutions. There's plenty. And so, in fact, they're cheaper than the US and they often work longer hours than in the US. And so the answer they propose in this document is AI and autonomous labs. Right. So we should move to a zone where the U.S. scientists are getting leverage from infrastructure, not their hands. And I think that's critical, not just in biotech, but basically across all of science.
And on the topic of AI, and obviously it
must be done. Like I really think it's worth people internalizing because we've forgotten what it's like to have a near peer competitor in science and technology, like since the fall of the Soviet Union.
Yeah.
And let me tell you, we have one.
Yeah.
And so then you have to look and see, where do we have competitive advantage and where do we not? And it's definitely not on cheap labor.
Yeah, for sure.
And so if we do science by the mechanism of cheap labor, we will lose.
Yeah.
Yeah. And so that, I do think it's time for a change.
Yeah, for sure. And so I mentioned at the beginning, obviously we're here for the AI Bio Summit at the NYSE. I mean, I'm here every day. You're here for the AI Bio Summit
I've been here today and 5 years ago. Exactly. Nearly. Yeah.
And so on the topic of AI, Ginkgo obviously recently collaborated with OpenAI.
Yes.
Bringing together advanced AI models and automated scientific experimentation. So away from the specifics of that collaboration for a second, just what excites you most about the idea of combining AI with real world scientific research?
Yeah. So, okay, if you think about, again for people who haven't done it, like how do you do science? Like what's going on, right? It's actually not that complicated. All right. You have a question and you basically plan an experiment that you think can answer that question. Mm-hmm. Then you conduct the experiment in the lab.
It's a scientific theory.
100%.
I learned about it in middle school.
Yeah. You get your result back.
You get your hypothesis.
Yeah.
You get your, come on.
And here's the part that they don't tell you in middle school, is that first experiment almost never answers your question. It gives you back a bunch of data.
I was told if I followed the scientific theory, I was always going to get it right.
That's good, answer it, and put on the poster board, right? You're going to get back a bunch of data, and it's going to make your problem more confusing.
Sure.
Okay, because now you've learned something about the real world that no one's ever asked before when you did your experiment. Does that make sense?
Yes.
And so you get that data back, and then you, as a smart scientist, look at the data, and here's the magic: interpret it, analyze it, and come up with a new experiment. And then you do it again and again and again. And so that part, like what I'm excited about on the AI side, I think the models are cool. Like we were talking about, like you were saying, could we understand, like, could the model have a cell live in it or a virtual self? Great. People should try it. It's not what I'm trying. What I'm trying to do is have the model do that interpretation and experimental planning.
Sure.
So you could just say, hey, here's my question. You plan the experiments, AI. You run the experiments and do that multiple times. And that's what we did with OpenAI.
And so I think one of the themes that emerged from that collaboration I was reading about is the idea that AI can move beyond simply analyzing information, as you said, and begin participating in that scientific process. You just mentioned that. How significant is it that AI systems are starting to contribute to experimentation? Like, what can this mean? This is something that you said you're doing. Yeah. We just talked about how it could, at a much faster pace, do these experiments so people just don't have to do it over and over again, right? Taking away that manual human labor. What can that mean when something like that can move from, I don't know how long it usually takes, probably weeks, months, if not years, to minutes and days?
Yeah. So the experiment we did with OpenAI, we let GPT-5 design 30,000 experiments a week for 6 weeks. Mm-hmm. We had a benchmark against this lab at Stanford, Mike Jewett's lab, who had produced a certain amount of, in this case, protein per dollar. It's like useful for manufacturing drugs and things like that. And we said, could you beat it? And after the 4th round, it had beat it. And after the 6th round, we beat it by 40%. And if it had said, hey, run water, like just put something pointless in here, we would have done it. We were doing whatever it asked. We weren't we were not designing the experiments. Okay, so that's a pretty impressive, like that's the first time I think people have really shown that the models had that capability. And that's actually a pretty old model. So we're doing some newer stuff now with them. But so that opens the door to, I think, a totally new way to be doing experimental science. And that has 2 impacts. One, just to give you a sense of scale, pharma industry spends close to $80 billion a year on research, not clinical trials, but just on labs and people in the labs and this type of stuff. The NIH spends $40 billion a year on the same stuff. So we're talking like $120 billion that's being spent largely on manual labs. All right. So that's my market. That's what I'm going after. I think that could be, you could get way more out of that $120 billion.
Sure.
And then the second question is, oh my God, what if we started to get a lot more out of that $120 billion, right? Like that's actually the much more interesting thing. Like, if I don't know, is it 100 times better? Does that mean we get 100 times more drugs? Right? Like what happens? Yeah. So I do think that's the question for science. I mean, we're starting to see this with, you might have noticed all this math stuff lately. Yeah. I just solved the Jacobian, whatever the hell thing I never heard of on Twitter, right? Like, and like, these are math people, but they're not like, they're not Terrence Tao, right? It's like a math grad student or something. And I think math it's hitting math first. I think every mathematician now believes it is going to have a massive impact at the speed that we learn in math. Why is math so easy? Like, why is it happening in math first, you know?
Just because it's numbers and they're able to sort of decode the numbers faster than they are able to the letters of a cell.
Yeah, yes, exactly right. In the sense that it all happens virtually, so you can like run the math problems.
Sure, cells are things.
I got to make them. And
cells are tangible things. That's a good point. Cells are tangible things. Math is I can do 1 + 1 on a piece of paper right now.
Right, exactly. And if you're in a computer, you can do it in a millisecond.
Yes.
And so that speed that it can learn, because it can try and see things that work. Its experimental rate is very fast because it's all virtual.
Sure.
Now, again, if I just point it out, most of the things we care about as human beings are not in the virtual world.
Yes.
Medicine, disease, food, blah, 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 longevity.
Tangible things.
They're all tangible things.
Yeah.
So if we want that, what you're feeling now in math to come into our lives in a way that isn't like just jamming more ads down our throat, it's going to have to be through the physical world. And so we got to solve these problems where you can now have rapid evals, like doing your tests very quickly so the models get better and smarter and can push the field forward, not in Jacobian whatever, but in Alzheimer's.
Yeah.
Right? And that requires labs.
So as we come towards the end of our conversation, if we're having this conversation again 10 years from now, and we're looking back at how science and research are done today, what are we going to say 10 years from now? What's going to make us say, why were we doing something that way? You know what I'm saying? Like,
yeah.
What are we going to be looking at 10 years from now that is going to make us say, like, what were we thinking?
So I'll talk about it from the level of like the practitioners, but then also like the average person, right? So I think for the practitioners, it's just going to be the lab. It's going to feel like the industry that has felt this the most viscerally is computer science. Like one of my co-founders is late 70s, this guy Tom Knight. He's an absolute computer genius, MIT professor. Got to MIT when he was 16 years old. He started at MIT in the era of like mainframes and punch cards.
Sure.
So he punched cards to make a computer program. Punched.
Yeah.
Okay. So he's
seen the evolution.
It's easy to look back and be like, wow, what the heck were we doing?
Yeah.
Right? Okay. Because we went on this journey. And so that automation journey, essentially getting our hands out of the lab and moving all that onto machines, I think scientists will look back and be like, wow, oh my God, can you believe we did that?
Yeah.
All right. Okay, so that's that. Well, let's talk about just like the average person. So I think there's some really interesting opportunities here. So look, we'll just talk about scientific method. All it is formalized curiosity, right? Like that's fundamentally what it is. And I believe like the one thing that makes humans unique among every other species on the planet, it's not actually like, oh, we're strategic or we had to plan wars or something. Actually, ants go to war. Yeah. Okay, right? Like there's other things.
That's the whole
hunter. Yeah. Tigers can hunt and
I feel like that's the whole theory of predator and prey.
Right, exactly. Yeah. All strategy and all this stuff. That's all there. What is unique about humans, in my opinion, is we're curious. Like that's what's fundamentally interesting about our brains is we're like, what? How does it work? How does it work? How does it work? And it like never. We're like, how does this literally, we're looking at like galaxies that'll have no bearing on our lives. We're just so curious, you know, right? Like that we want to know how it works. Okay. And so I think that's like in everybody. And we're not able today, like the average person has a question. And unless they can like Google the answer, if it's a new thing that like no one has found out before, there's nothing they can do about it. They don't get to do science. And I think AI models can turn your question into a well-planned experiment. And then I swear to God, you will someday just order it up and be like, hey, what's going on with this? And I think the first place you'll see that is in your health. Like health and wellness, people have an endless list of stuff about them and their thing. And we're all different. My genome's different than yours, but the medical system basically treats us all the same and the drug companies treat us all the same. And so like, why can't I start to have more information about myself? Like what you see now with like wearables and that kind of trend, I'm telling you, we'll look back 10 years from now and you'll be like, can you believe all I knew was like how long I slept last night? I didn't have
my
like
sleep score,
a regular readout of like all my biomarkers across my body at all. You know, like you're going to be blown away, I think, by where that goes, because now we'll have this interpreter who can let you ask questions about yourself. And I think that'll be where it begins. And then who the hell knows where it goes, right? If everyone's a scientist, it's an interesting world.
It definitely is an interesting world. Well, Jason, I really enjoyed the conversation. Thank you so much for joining us inside the Ice House.
Yeah, thanks for having me, Lance.